Dear students, in this unit we will talk about the life cycle of Rhizopus. Now, Rhizopus is a famous genus of fungi and that comprises of saprophytic as well as parasitic fungi. Now, as you are aware that saprophytic fungi are the fungi which lives on the dead animals or plants or organic matters, while parasitic fungi are those which lives on living animals, plants, or humans. Now, they actually are the member of class Zygomycetes. Now, when we talk about their habitat, they are mostly found in the moist and damp places, and they are commonly found on organic substances like vegetables, fruits, breads, jellies, etc. So most of the kitchen uh, things or the vegetables or fruits, they are wasted by this Rhizopus because they infect those bread, jellies, and plants and vegetables, and they infect on that one, and they result in the decaying of these organic matters. Now, when we talk about the structure of Rhizopus, its vegetative structure is actually made up of cynocytic hyphae. Cynocytic means they don't have septa in them, and there are more than one. They are actually multinucleated hyphae. They are present in them, and most of them they are branched in structures. They are not in the form of a single structure. Now, when we talk about the reproductive cycle of Rhizopus, they contains both sexual as well as asexual reproduction. As you can see here, their asexual reproduction takes place when they form sporangiospores and chlamydiospores. Now, in these sporangiospores, as you can see here, that a single mycelium gave rise to a structure which contains sporangia. So, sporangia is actually is a structure which carries a large number of spores which can be dispersed into the environment, and they can give rise to different hyphae and mycelia whenever they found some favorable conditions. But when we talk about the sexual reproduction, in sexual reproduction, what happens that two hyphae from different mycelia, they come close to each other, and uh, they for example, just like we see that one mating type we can call as plus, and the other mating type we can call as minus. Now these two hyphae come close to each other, and they make a structure which known as gametangia. Now these gametangia carries haploid nuclei from both mating hyphae. Now, after the gametangia, the mixing of their cytoplasm takes place, which is known as plasmogamy. After plasmogamy, their karyogamy takes place, which means that a structure which is known as zygosporangium, this is synthesized. It's a very hard structure. You can see the actual structure of this one. It's a very hard and protective structure which is dispersed into the soil, and it contains many diploid nuclei, and at the time of when there is a suitable condition, there is moist as well as damp conditions, this karyogamy, this zygosporangia, it develops into a sporangium, and this sporangium then contains many spores which are dispersed into the soil, and after finding the favorable condition, they can grow, and they can form independent hyphae and mycelia. Now, during the asexual reproduction, the sporangiophores, as you have seen in the diagram, it contains a lot of sporangias, and mycelium directly transfers these nuclei into this sporangium, and as a result, there are a number of diploid spores are formed, which are dispersed through this sporangiophores. They also multiply through vegetative reproduction, in which what happens that a mycelium or a hyphae can break at a point, and it results in the formation of two different hyphae, which can independently grow to form a complete a structure of a fungi.